So without further ado, it's my great pleasure to introduce our speaker, Dr. Marta Gomez-Chiarri. She is an aquatic pathologist. She coordinates the Interdisciplinary Sustainable Agriculture and Food Systems undergraduate program, a major that explores the food chain from farm to plate to waste and back, emphasizing sustainability, impacts on human health, and resilience from economic, environmental, and societal viewpoints. She is also the coordinator of graduate programs in the Department of Fisheries, Animal and Veterinary Science at the University of Rhode Island. Marta's research interests include the use of multidisciplinary approaches to the prevention and management of diseases in marine organisms, from probiotics and microbial interactions to genomics and comparative immunology. Her collaborative national and international research on marine diseases is driven by a desire to ensure equitable access to healthy food that is sustainably produced. So without further ado, Marta, I'll turn the presentation time over to you. Thank you.
Thank you, Nick. Thank you very much for the nice introduction. And also I'm looking forward to sharing our research with you. It's nice to see some familiar names on the list of participants. And Nick, thank you so much for inviting me. And I also want to thank the rest of the team that organizes the seminar series. This is a great honor. So before I get started, I want to actually acknowledge the people that have done the work. I write the proposals and I help with the papers, and I provide advice. But as you well know, graduate and undergraduate students, and every once in a while I have a postdoc, are the ones to do the work. And so here you have the current team in the lab. Jessica is a PhD candidate on her second year. Evelyn is also a PhD candidate on the third year. Rob is in his first year, also a PhD candidate. Everybody is a PhD candidate in this, in the lab right now. Jamal is also a first year PhD. Erin is almost finishing. She'll defend in July. And Tejasri defended last year, and she's now, after being 2 years ago, and she was a postdoctoral fellow and now working in industry. And Rebecca defended a year ago, and she is a postdoctoral fellow at the Pasteur Institute in France. I also have great collaborators. David Nelson is a microbial physiologist at URI, and David Rowley is a natural products chemist. And this is just the collaborating team for the research that I'm going to talk about today. This is kind of an overview of my research with the agencies that have funded it. Nick mentioned that it would be great if I gave you a little history of my career. I started as a biochemist and molecular biologist for my bachelor's in Spain. I also did my PhD in Spain in immunopathology, but focus on biomedical, human disease. I work with rats and mice, but I always wanted to work. Basically, I wanted to live in a coast town, and I wanted to work on aquatic organisms. So, I did a postdoctoral fellowship at Stanford University, Hopkins Station, thanks to a fellowship from NATO, which does fund research, or used to. And I was working on abalone transgenics there. And then I moved to work on the design of vaccines for finfish, work on salmonid vaccines. And after almost 5 years at Monterey in California at Hopkins, I applied for a job at URI in the East Coast and moved there in '97. And I've been at URI since '97. I haven't gone, besides a sabbatical in Seattle and another one, I think that I haven't gone very far. So my research is, I like to go from fundamental basic science to application for my research. And it starts with identifying, involves either identifying pathogens, developing tools for monitoring, looking at host microbial pathogen interactions, and how to manage disease by taking all the knowledge that there is about the impact of the environment, those interactions, to develop management tools. And the ones that I've been going to be talking about today are basically these two in the right side of the slide, the mechanisms of disease resistance and also using probiotic. And I'm going to focus on oysters because oysters is kind of the only aquaculture industry in the state of Rhode Island, and it's the one that has been taking over all my research in the last years. Although I've also worked in many other marine organisms and also freshwater. So I don't know how much you know about oysters, but oysters are pretty important both ecologically and economically, and I'm not going to talk too much about them as their importance, but I also wanted to tell you that we are working on diseases that affect all stages of oyster production and oyster life history in the ocean, and from larvae to adults. And so we have diseases that affect the larvae, and major ones that I've been working on. I don't work in oyster herpes virus, which is mostly a disease of Pacific oysters. But for the American or Eastern oyster, and for all oysters, vibriosis is a huge problem that really impacts production in hatcheries. And so that's one of the diseases I work on. I also work in some other diseases, bacterial diseases. This disease is called juvenile oyster disease, and it only impacts oysters in the northeast of the United States. And then we have several parasitic diseases that affect oysters as seed or adults. And these are Perkinsus marinus, which is a protozoan parasite that causes a disease called dermal disease. And then we have some Clostridium parasites that cause MSX and SSO diseases. And so I'm going to start with talking to you about probiotics and the research we've been doing for the last probably a little more than a decade on this. And this has been a highly collaborative research with a lot of people involved. This is through the years. And it goes from those basic mechanisms of action to implementing probiotics at the commercial scale with companies and developing formulations to be able to use them. And probiotics is one of those things that, sometimes they work, sometimes they don't. And we really want to know how they work and why, what are the conditions they work in so we can improve them and make them more reliable. So we're working mainly with two probiotics. One is a Phaeobacter. It's gram-negative. And the strain that we work is S4, and the other one is a Bacillus, which is a gram-positive. And both have been isolated from local waters in Rhode Island. So they are ubiquitous, they are pretty common in marine environments. And they both produce antibiotics. They are different antibiotics, and they both work by different mechanisms of action. That's what we found through our research. So we've started by isolating the probiotics thanks to their antibiotic activity against the vibrios that we care about that cause disease in oyster larvae. And we do then trials with the oyster larvae in the lab, and then we transfer them into the hatchery, and we test them. Once we have activity in the hatchery, then we tried with other bivalves, and we discovered that they work on base scallops and a little bit on clams, but they don't work on mussels. So there's definitely a difference between the host where you can get the disease to work, even if the vibrio affects all this larvae of all the species equally, or more or less equally. Then we develop formulations. Basically, that's a way in which we can, because these probiotics have to be given every day in the hatchery to really have good activity, to consistently work. So we need to develop something so you don't have to grow them fresh, because they are live organisms that don't have any activity as killed organisms. So you have to have something that keeps them alive for a while. and that the hatchery managers can provide, can go to the fridge, take a sample, and then mix them with the food and give it to the larvae. So we developed that, and we've been testing those in the hatcheries to see how they work. We've also looked at mechanisms of action, and I'll talk about one of those mechanisms of action today, which is immunomodulation of the immune responses of the larvae. But we also look at how the probiotics affect the microbial community of the hatchery, and many other things. So one of the things that we discover, and I won't touch too much on this, besides telling you that, and this is published, and here are two of the publications. There's another one coming up in a little bit, in a few months, that the mechanisms of action of these probiotics are very complex, and they're different depending on the microbial species. The bacillus is different from the Phaeobacter. But we know that if we knocked out the genes that regulate biofilm production in S4, it loses about 40% of the activity. If we knock down quorum sensing, it knocks down 40% of the activity too. If you knock down both, you inhibit about 80% of the activity. So they seem to be, and also we know that type 6 secretion system has a role in these interactions. And we were wondering if immunomodulation was one of those mechanisms that allow this probiotics to work. So Tejashri, while she was a PhD student in the lab, looked at the immune responses of larval oysters to the two probiotics as compared to the pathogen. And this was just published in 2020 in an issue of Vaccines. But what she did basically is she took larvae and she did 3 different experiments from larvae from different hatcheries. They exposed them, and she exposed them to probiotics. So usually what we do is we expose the larvae to probiotics for 24 hours, and then we provide the pathogen. Because it has to be a pre-exposure with the probiotic. We have to allow it to get established before we can get protection. So she exposed the larvae to the probiotic, and then she added the pathogen and looked at the immune responses. In here, I present the individual immune responses to the probiotic or the pathogen, not everything together. But at the same time, so you can see how the larvae really responds very differently to either 3 of those microbial, the pathogen or the 2 probiotics. In 6 hours, this disease is really quick. So you put the pathogen with the larvae, 10 to the 5 CFUs per ml, and in 6 hours, you don't have mortality, but by 9 hours, they're starting to crash, and by 20 hours, they are all dead, or most of them are dead. In this case, 40%, because we kind of target the challenge dose so we don't do an overkill. And so when we're looking at 6 hours is before mortality. So we're looking at the gene expression of the larvae before mortality. And we decided to use the same time for the probiotics to compare them equally. So I'm going to show you the results from the gene expression of the larvae in response to the probiotics or the pathogen using volcano plots. And just in case you're not familiar with volcano plots, they're going to look like this. And so each dot in this graph is one gene, the expression of one gene. And in the x-axis, you have levels of gene expression with 0 being here in the middle, where there's this is no expression. And so if you go to the right, you see upregulation of gene expression. So here is a lot of expression. These genes in this quadrant in here are highly upregulated compared to the control. The ones in this side are highly downregulated in compared to control. And then the y-axis is significance of that value. So the p-value basically. And so this is a log scale in both areas. And so anything that is up here is highly downregulated and very significant. Anything up here is highly upregulated and very significant. So, and actually I forgot that I had an animation for this. So So Tejas really did that, and this is the pathogen RE22 at 6 hours. The program arrived bacillus at 6 hours. This is as far as 6 hours. And just by looking at these 3 images, you can see that they are very different volcano plots. Some are flatter, some are higher. But and I'll highlight this, I'll blow it up a little bit so you can see what I mean. But I can give you the preview of what we found is that all these patterns of gene expression indicate that the pathogen RE22 immunosuppresses the larvae, while the probions provide immunostimulation compared to the controls. So, so these are, and I'll show you more about it. So this is what indicates that there's a lot of upregulation of immune gene expression, while this in here, this yellow area will show you that there's downregulating by the pathogen. So some of the common transcripts that are upregulated to both probiotics are involved in inflammation, recognition of bacterial signatures, and some leptins. And these are traditionally seen in response to bacterial infection in invertebrates and also in vertebrates. So you can see some of these genes in here. So the caspase-1 is one of the ones that most significant and most highly upregulated. You can see it here, caspase. And that was mostly in here. It's also with the pathogen, but you can see it for both probiotics. And you can see basically a lot of other immune genes that are upregulated in response to both the probiotics. And you can see that there are some common ones, but they're pretty different. They're not exactly the same. So then for downregulation, what you can see in here is, and these are not, very downregulated, are a lot of genes that are involved in signaling pathways. The MyD88 regulates signaling pathways that will then lead to the expression of immune genes. So the pathogen in some way is downregulating those signaling pathways that allow larvae to respond to the pathogen. So we found very interesting. So these are some unique genes. And this is kind of a summary of what we've seen. So the RE22 downregulates this NF-kappa B, MyD88 immune pathways, while the probions upregulate them. So what is happening, we think that helps the larvae, is because we are providing those probions 20 hours before the pathogen. The larvae are responding to the probions, and they are expressing all these effectors that will kill, the RE22. But RE22, if you don't have the probions, RE22 is able to inhibit the signaling pathways, so the effectors are not there anymore, and the RE22 is able to kill the larvae. So by exposing, basically you're providing, and the probiotics are being eliminated by the larvae. They don't stay, but they are stimulating that immune response, so the pathogen cannot do that even if this downregulating the pathways, then the effectors are there, and in combination with the antibiotic activity and all the other rules, that helps eliminate the Vibrio from the system. So we were really interested on that. We also wanted to see if there's anything else that is happening that we are not capturing. So Jessica is now looking at that interaction between RE22, the pathogen, and the probiotic in competition using transcriptomics. And I wanted to talk a little bit about this. I'm going to go pretty quick, but we know that the Vibrio has many virulence factors that contribute to killing the larvae. And one of them that we're really interested lately is the type 6 secretion systems, which are basically, think about it as an injection system that the pathogen uses to fight either other bacteria, so is involved, or to kill larvae. And so there's in this vibrio that we use, RE22, there are two type 6 secretion systems, 1 and 2. And basically it's kind of like, you know, you have a barrel, a needle, and a tip, and that are used to basically inject effectors that kill into either bacteria or eukaryotic systems. And this is another, basically. So all bacteria, many bacteria have this, and usually they can also inject stuff into their own, you know, peers, their own congeners. But what those bacteria, they basically have is antitoxins to whatever toxins are infected. They have the protection mechanisms, so they don't kill each other. And some of those mechanisms are kind of things, antitoxins or really good efflux genes that are able to pump those toxins out. So, so what we did first is we knocked out those type 1 secretion systems, either the tip or the barrel, in RE22. We tested them against larvae, and this is basically the survival of the larvae after a challenge. So the control, good survival with the RE22 wild type, you have about 50% survival. When you knock down either the type 6 secretion 1, you completely eliminate that ability of RE22 to kill larvae. When you knock out the type secretion system 2, they still able to kill the larvae somewhat. And so what we know is that type 6 secretion 1 is mostly used by RE22 to kill eukaryotic cells, while the type 2 is mostly used to kill other cells like the S4. Jessica did the competition experiments between the two, and you can see that when RE22 is in monoculture, it is basically grows to 10 to the 6 in a biofilm. When RE22 is with S4, it just really knocks it down. Okay, so Jessica wanted to see what other genes and what other functions besides orange quenching and TDA production, the antibiotic production by S4, are involved in killing that RE22 or immunite. So she looked first at the RE22. I'm going to present that. And then she just basically put 24 hours the S4, then 24 hours added the RE22 to the dish, waited for 5 hours, and then collected the supernatant and the biofilm and look at gene expression. And we are seeing what are these two bacteria using to fight each other. So this is the same kind of plots that we did for oyster larvae, but in this case, we are looking at gene expression by the bacteria by RE22. And so what did Jessica found is that when RE22 is cultured with the probiotic in competition, the probiotic leads to downregulation of those type 6 secretion systems. They are these green dots. This is a bunch of genes involved in the type 6 secretion system from RE22. So S4 is able to downregulate those virulent factors from RE22, which was really interesting. And RE22 is trying to defend itself because what it's upregulated in compared to control when present with S4 is all these efflux genes, very upregulated. So RE22 is trying to protect itself from the S4 attack. And this is both in biofilm and the supernatant. So you can see that those genes are expressed. And these are some of the highlights of what Jessica was seeing. So we know that S4, the probiotic, is able to prevent infection by RE22 in larvae. And we know that things that contribute to that is, so we know that RE22 can kill larvae, using many different virulence factors, including type 6 secretion systems and immunosuppression, while S4 is able to prevent RE22 for having that effect by stimulating an immune response in larvae, but also by regulating the production of virulence factors by RE22. So it's a complex system, but very interesting. So I'm going to switch gears now. So that was one part of the story about probiotics. So probiotics work great to prevent disease by Vibrios in the hatchery. You cannot really give them in a grow out oyster farm, which is done, in the environment. It's done in our coastal estuaries. So we're looking at exploring other ways to prevent disease in oyster grow out, and that's through selective breeding. And there's a East Coast Shellfish Breeding Consortium. There's also several breeding consortiums that target different species, but we have a big one that is focusing on using genomic selection, to enhance breeding for disease resistance. So both Tejasri as a postdoc and Erin in here, have been working on this project or with the Eastern Oyster Genome Consortium. So one thing that I can tell you is that the breeding consortium have several challenges because the diseases are not in the environmental conditions that oysters are exposed through the range are very different from the Northeast all the way to the Gulf. And the diseases, all the diseases are present in the Northeast, but, only dermo is present right now in the Gulf. So one of the things that we've done as a consortium is first sequence the genome of the Eastern oyster. And the sequence of the Pacific oyster was already available. And one of the things that were first seen when all these molluscan genomes were being sequenced is that oysters and other mollusks have many more versions of immune genes involved in recognition and signaling than other species. So it seems like they expand the number of genes. This is one way that they may use to respond to the diversity of pathogens out there. Oysters don't have an adaptive immune system with antibodies, so they have to deal with diversity of pathogens in a different way. And they are doing it by expanding certain gene families. And some of them are like pattern recognition receptors, like toll-like receptors up here, signaling molecules like MyD88. There's a lot of different genes. So my lab is interested in some of those gene expansion and the potential role that they have in immunity. And one of the functions that we're really interested in is apoptosis, or regulated cell death, cell suicide. And apoptosis is critical for many different functions, not just immunity. And it's basically a cell, when it's damaged, can choose to commit suicide. So, doesn't affect the whole, physiology of the organism. And so cells that are infected by viruses or other intracellular pathogens, and RE22 has also an intracellular lifestyle from what we know, can choose to commit apoptosis, so the pathogen cannot proliferate within the cell. So Erin was interested in figuring out what is the role of apoptosis in response to many different pathogenic challenges. And she also was interested because one of the things that we saw in the genome is that a family of genes that regulates apoptosis, called the inhibitor of apoptosis proteins, are very expanded in mollusks. And these numbers are kind of hard to see. I have to tell Erin to change the color. I'm an old person, so I just have a hard time with these things. But so this is a tree of mollusks. And so we have octopus at the bottom here, cephalopods. Then we have some bivalves in here. a scallop, and two oysters. And then we have gastropods up here. And you can see that the number of inhibitors apoptosis is very different depending on the species. And I can tell you, in humans, there are 6. And so octopus have 10 and 11. And then you have something like the snail that transmits schistosomiasis has 88. And Eastern and Pacific oysters, it's interesting. They have about the same size genome, but the Pacific oyster has 40 and the Eastern oyster has 70 or 69. And when you look at the chromosomes, this is representation of the chromosomes in the oyster. This is kind of the chromosome map, from chromosomes 1 to 10. You can see that the versions of this inhibitor apoptosis proteins, which are this little line, are mostly located in 2 chromosomes, chromosome 6 and chromosome 7. And they are in tandem. So what has happened is this family of apoptosis regulators has expanded in oysters through tandem duplications. And those tandem duplications they stay in the genome, probably because of selective pressure due to different environmental stressors. That's our hypothesis. So this is another view of that expansion of the genes. And you can see red, these are all those snail versions, and these are the oyster versions. And you can see that these are truly expansions that occur within one gene and within one species. So they are not expanded throughout they are just really complex history of gains of genes and losses of genes in different species. And you can see here, you have, I tried to show, I'll show you in the next slide, but we have gastropods and mollusks in here. And you can see this is really complex. The genes are all over the place, but there's some genes in the inhibitor of apoptosis that are only present in gastropods, and some versions that are only present in oysters. And some that are only present in bivalves. And so Erin looked at all those genes that were expanded, all those new versions, and characterizes their domain structure. And what she saw is that we have novel types of inhibitor of apoptosis that have not been described in any other species. And some of those you can see in here, like this is one example of a novel type that has, and she has characterized those domain structures to kind of try to have an idea of what their function may be based on the functional domains. And I won't go into that. We're sending this out, I promise, this month. And so hopefully you'll be able to read the paper in this year. So Erin looked at the gene, so in this tree of inhibitor of apoptosis, there are only 3 species, scallops and the 2 oyster species. And these are the gene expression levels of all these genes in response to pathogenic challenge, or actually microbial challenge. And so we have the 2 species. We have in B is the Eastern oyster, and in C, in this block here, is the Pacific oyster. And where you see a line, when you see a color, is that gene is expressed. Actually, these are transcripts, not genes. Some of them are basically same gene with alternative splicing, but it's very complex. But what I want you to get out of here is these are bacterial, both in the Eastern oyster and the Pacific oyster. And this is a parasitic challenge in the Eastern oyster, and these are viral challenges in the Pacific oyster. We don't have a Pacific challenge. These are transcriptomes that were available in the database. We don't have a Pacific challenge, a viral challenge for Eastern oysters because it hasn't been, they've been recently done, but by the Friedman group and Colleen Barch, but those transcriptomes, there are no transcriptomes available. And there's no parasitic challenge on the other side because dermal disease is not present, has never been described in the Pacific oyster. So you can see that the patterns are very different between the two oysters, but not all genes are expressed to the challenge. And it looks like for each cluster of genes, for each gene type, there's only maybe one transcript that is being expressed to a particular challenge. And that's especially true in the eastern oyster. In the Pacific oyster, there seem to be more, the ones that are expressed are more commonly shared between different challenges. And some of the novel types of, these are some of the novel types of IEPs that have not been seen in other model species. And you can see that they are being expressed. So these genes that are expanded are being used in response to immune challenge. But some are only with bacteria and viruses, not with parasitic, and some are only with the parasitic. Actually it's harder to see some that are only expressed with the parasite, but it's still a very complex history that we're trying to tease out. So what Erin did is she looked at which other genes, immune genes, are co-regulated with inhibitors to apoptosis, and by doing something called a network analysis. And so these are, you're not going to be able to read this, but these are only genes that are involved in apoptosis, because one possibility is that once those inhibitor apoptosis genes expanded, the new version that gets created by a duplication may have a different function. So we were wondering, are they involved in immunity still? And what we saw is that they still co-regulated with other immune genes and also with genes of apoptosis. So we know that they are involved in immunity and they are involved in apoptosis, most likely because they are co-regulated. We still need functional studies. But one interesting thing is that we were trying to see if like particular members were associated with particular types of pathogens. And that didn't come up through for all the pathogens that we used. But interestingly so, when you do a clustering of the gene expression for the immune apoptosis genes and the inhibitor of apoptosis, they don't cluster necessarily by species, Virginica versus Gigas. So you see CV is Virginica, the eastern oyster, and CG is the Gigas. Or by bacterial or parasitic challenge, but they are very different depending on the resistant, because in those experiments, there were oysters that were, in some cases, some of these experiments, we had some oysters that were resistant to the challenge and some oysters that were susceptible to the challenge. And you can see the one that shows a very different pattern of gene expression for these genes is one that is susceptible to bacterial infection in here. And the other ones that are, and that's the same thing happens for the viral. The susceptible family that is susceptible to the oyster herpes virus shows very different pattern of expression than the resistant. So that indicates that these genes may have a role in resistant or susceptibility to the disease. And that's something that we are exploring more with more challenge experiments. And that will be in Erin's defense in July. And hopefully later in the year, I can show you more about it or it will be published. So in summary, we are looking at immune responses of oysters to many different pathogens. And also we compare patterns of gene expression between resistant and susceptible oysters. And what we see is that that mollusks have more versions of certain immune genes and genes involved in apoptosis than many other species. And that the patterns of expression of those genes in response to challenge seem to vary depending if they are resistant or susceptible. Not necessarily by the pathogen, but more with the levels of susceptibility. And that's really interesting. So Erin, what is doing right now is relating this gene expression patterns with apoptosis and with the Perkinses, the pathogen with the protozoan parasite that causes dermal disease. And what the Eastern Oyster Genome Consortium is doing in the breeding consortium, we have now genetic markers for these genes and many other genes, and we're looking at patterns we're seeing if those particular gene variants for many of those genes are associated with resistance or susceptibility using a SNP array. So we're trying to link the phenotype of oysters for many of these immune genes with the genotype of the oyster by having resistant and susceptible oysters. That will allow us in the future to identify potentially markers that may be associated with disease resistance. So I went pretty quick around this, so we'll have a lot of time for questions, but what is next? So one thing that Rebecca, one of the students that graduated, I have also a lot of data about how the probiotics affect the microbiome, the microbial communities, the bacterial communities mainly, of oysters upon treatment, how the pathogen affects those microbial communities. And Jessica is going to look at how the interaction between the probiont and the pathogen changes in the host. So we are going to have transcriptomes of all the pathogens, the probionts, and the larvae host all together. And we're trying to find ways to analyze all that complex data together. And then we are going to treat if and we know that the probiotics don't work very well in some conditions, and it could be that there's other players, other microbes that inhibit their activity. So by looking at the microbial community of the oysters from many of the hatchery trials that we're doing, we're trying to identify which other members of the community could prevent probiotics from working well. What else are we doing? And then we're doing all that genomic selection tool development that I told you in the previous slide. So this is basically, I'm going back to the acknowledgement slide by showing the different agencies that have funded the work. I already showed you who was involved in each of which students was involved in each of the different areas that I've discussed. And I love to answer any questions you may have about this research. And and feel free, like Nick said, if to turn on your camera and unmute yourself to ask questions, or you can put them in the chat. And looking forward to having a discussion. We have plenty of time. And I can go back to particular slides if you want me to clarify, since I understand I've gone pretty fast. So there's time for that now. Thank you.
Thank you very much, Marta, for excellent presentation. Lots of good work, very interesting stuff.
So I cannot see the chat. Let me see.
Nothing in the chat box yet, but.
Okay. So
okay.
if you can check it for me, because I forgot to put it. Let me see. I think I can get it.
We got one here for you from Gary Marty. He asks, when looking at the gene expression of larvae that are infected with bacteria, how is oyster gene expression separated from bacteria gene expression? Both genes are in the same sample. How are the genes of each sufficiently different?
Yeah, so that's a great question. So in the transcriptomes, we use total RNA. So, pathogens are there. You can, all the pathogens, many different things are contributing to that gene expression. So what we did is, that's why we made a lot of effort to sequence the oyster genome, to have that sequence of the oyster genome. And we also have the whole genome sequences of the pathogen and the probiotics. So what we definitely do is we map the reads from the transcriptomes to each one of those genomes. And so they're sufficiently different that if I read, they map, as you map them, you get the ones that, map to the oyster, you analyze them as oyster, the ones that map to the pathogen and the probiotic, and with that, we use a certain threshold we know that really differentiates them by comparing to other species. And that allows you to separate the patterns of gene expression. And then you can, because the number of genomes that are available in the database, it keeps growing, you can identify many other things by mapping them to the whole, to the genome data sets that requires a lot of computer power. It takes a long time. It's usually, we have a cluster with a lot of nodes, and that is the most time consuming part of the whole analysis process, which could take, if you don't have enough computer power, it can take like a week, but usually we can do it in a day. Yeah, we do perform a ribosomal RNA reduction. So we have, depending on the kit that we're using for sequencing, is usually, this is all done with Illumina. There's a few ribosomal kits that will reduce ribosomal that are, we've tried different ones. There are different ones that are better for bacteria versus different, invertebrates versus, and so we use those kits to reduce the ribosomal contamination. There's still some, but we reduce them. Yeah. So the software that we use to map is, so we have a pipeline, and I don't have the slide here that shows the pipeline. We've used different methods. One of them is BWA, is basically their read mapping software, and it just basically looks at sequence identity in different areas, and then provides a quality score that tells you what is the likelihood that it was kind of assembling like a puzzle. I can send you the link of the GitHub where we have the whole pipeline for transcriptome analysis. That's already available. It's open software. So let me make sure that I should have gotten it ready for you so I can put it in the chat, but why don't you email me? I'm going to put my email in the chat and remind me to send you that. Okay, no problem.
It looks like we have a question from Esteban. If you want to go ahead and.
Hola, Esteban.
Hola, Marta. Excelente.
Gracias.
Always a pleasure. Marta, I actually have two questions for you, and one of them, is very general on the immune response of oysters. My understanding is that oysters have an open type, circulatory system, right? And that the hemolymph
absolutely.
And that the hemolymph is not sterile, right? Like, you can recover, all sorts of bacteria all of the time. So my question is, when you do all of these gene expressions, transcriptomic analysis, particularly during the challenges, how consistent, I know you use controls all of the time, but I was just wondering, how stable is that gene expression since they're already seeing, all sorts of bugs, all of the time. I know that you have to compare it with the controls, but how replicate, how easy it is to replicate, when you use oysters, outside, for example, in the open systems.
You are correct that this is a big challenge. So I showed you in here only the most consistent signals that came from so that's why we use a lot of replication, because each oyster in a population, even if they are from the same family and related family, show very different patterns of gene expression and have very different microbial communities, amazingly so. Even oysters that are living next to each other in the environment show very different microbial communities. I mean, they have a lot of commonality too, but they show a lot of difference too. So there is a huge level of variability in these experiments. So I show you that Gomez-Chiarri did 3 different experiments. So we have both technical replicates for sequencing, which each sample from each oyster is sequenced 3 times. And then we have biological replicates, which are oysters within an experiment. And then we repeat those experiments like we had whatever number of oysters. Actually, usually with larvae is larval pools, thousands of larvae. But we realized that in order to get significant patterns of expression, we need at least 10 oysters sometimes to get enough, to be able to differentiate two groups, just because and part of it is that microbial community that is affecting this gene expression. And then Gomez-Chiarri did 3 different experiments with larvae from 3 different hatcheries, and it was all over the place. But there are some consistent messages from it. And what you saw that I presented is the thing that was consistent between the 3 different experiments.
Very cool. Yeah, it's always a challenge, I'm guessing. And
It is crazy.
The other question, Marta, that I had was in one of your slides, I think that it was mentioned that toll-like receptor 4 was upregulated in to the probiotics, and I was just curious, because TLR4, at least with fish, my understanding is that just a few of them have it and or have been described. And really, it works, very different in the recognition of LPS than in mammals. So I was just curious in oysters, how specific it is to the LPS, because I think I saw that one of the bacillus, the probiotics had also an upregulation. So I was just curious in general for the TLR4 in mollusk or in oysters, how well described is that, and is it specific for LPS?
That's a great question. There's some work in the Pacific oysters about characterizing TLR4, but I don't think it's as specific. There's not much known functionally. So, when this is just a hypothesis, at this point, we haven't done the functional studies in Eastern oysters to know if it's just responding to LPS or is responding to a broader pattern. The interesting thing about those 2 probiotics is that even they did stimulate a lot of expression of different TLRs. So it was 4, 13, but it was a bunch of them, and there's many different versions of those TLRs in oysters. So why do they have even many different versions of TLR4? I mean, do they all respond to LPS? That's an excellent question. That would be a great experiment to do. And we have to develop the tools to do that functional assays. We still have to develop CRISPR, and oysters are difficult to transform, to genetically introduce DNA in them because but I mean, people are working on developing those tools so we can investigate those functions a little bit more. But yeah, that's a great question. And another thing, the probiotics did stimulate immune responses consistent with a viral response too. So they may be crosstalk between all those different pathways. Thank you.
Excellent presentation. Thanks very much for that. Quick question, a little bit about using the probiotics in the hatchery with the oysters. If you're applying a probiotic daily, it looks like it's causing an upregulation in these immune genes. With some animals, if you constantly stimulate their immune response over time, that immune response can crash. So is there a certain limit to you don't apply these for more than 2 weeks or something? Do you know if there's a time frame where you would expect that immune system's not going to be as upregulated anymore if you're constantly using these? Yeah.
We need to do a little bit more on that. But first, the larval, the hatchery stage in the hatchery for oysters is about 15 days. So 15, we usually are treating them for 12 days. 12 to 14 days, and then they develop, they settle, and what we've seen is actually, while you need to keep providing the probiotic as larvae, it seems like a seed you only need to do it like once, or they have a longer, you don't need to keep going it. But we've never really done much. We're just starting to do some of those experiments because it's not really commercially relevant to give the probiotics after the seed stage. We're just looking at after we stop giving them when the larvae settle, and they're seed, how long would that immune response last? And we just collected those samples this hatchery when we're doing we're doing the experiment right now. Evelyn, which is one of the students, is doing that, collecting all the samples right now. So hopefully I can tell you more in a year from now.
Great, thanks. Yeah, it sounds like since that hatchery stage is so short that maybe you don't get to that point where you're overstimulating their immune system.
Yeah, and there's great research being done at Oregon State University with other probiotics in Pacific oysters. And actually they are mixing, I saw this at the National Shellfishery Association, they are mixing, we are only doing a single probiotic for this mechanism of action to get to know more, but when they mix 10 probiotics that don't fight each other, because as you saw, these bacteria keep fighting each other, they get a longer term protection. They don't need to keep giving the probiotics as often. So that could be a really interesting avenue, just, you know, providing complement, you know, I think this maybe it's because of different mechanisms, not just immunostimulation. So
Sure. Thanks.
thank you. Great questions.
All right, we've got time for one more question if anybody has one out there.
And if anything comes up after, you know where to find me. So thanks, everybody.
All right, well, thank you very much, Marta, for the amazing presentation and all the awesome work you shared with us today.
Thank you.